Hey guys, welcome back. So today we're going to be talking about our new unit on heredity. So if you want to read your book beforehand to kind of give you a kind of preview of what we're going to be talking about, that might be a really good idea. It's in Cells and Heredity, Chapter 3, Lessons 1 and 2. Also, you can read it after we go through today's PowerPoint, whatever's better for you. If you have any questions, obviously feel free to reach out via email or posting it to the discussion board, however you feel comfortable asking your questions. So let's get started. All right, so what is heredity? Heredity's definition is it's the passing of physical characteristics from parents to offspring. So, a trait is what we're going to be calling those specific characteristics that can be passed to the offspring. So, for example, the color of a flower, the height of a person or a plant, your eye color, your hair color, your shoe size. Those physical traits are going to be what we're talking about. Again, those are coming from genes. And what are genes? A gene is a sequence of DNA that determines the trait. Because remember, our DNA are the instructions for our bodies. So the gene is just one little section that gives instructions for one specific trait. So this DNA, again, passed from parent to offspring. When we have different forms of a gene, we call them alleles. So a gene, again, that section of instructions from the DNA, controls the trait of the height of a plant. So we have the instructions, again, those genes, indicating the height of a plant. One allele, so one version of that gene, is for short plants. Another allele, another version of the gene, is for tall plants. So we can have multiple different alleles for one specific trait. In this example, we have two different versions, two different alleles, short and tall. So, we all got to get our traits and our genes from somewhere. Humans, most animals, even plants, we've talked about pollen, inherit two alleles for each trait. One coming from mom, one coming from dad. So, one coming from the egg, one coming from the sperm. These alleles that are inherited control what your traits are. So, everything coming from mom and dad determines who you are physically. A dominant allele is one whose trait always shows up if that allele is present. So let's talk about that. Dominant. When we hear the word dominant, we might think of powerful. We might think of controlling, which is very fitting for this name. A dominant allele always wins that battle, always wins that fight. If that allele, if that version is there, that's the one that you're going to see. A recessive allele, on the other hand, more laid back, hangs back. If both inherited copies, so both alleles that you get from mom and dad, are recessive, they're going to then be able to be seen. But if you only have one of them, if you only have one recessive allele, then it's going to be hidden by that dominant allele. That dominant allele, those are the instructions that are going to be read and created and performed. But the recessive allele is going to be there. It's just not going to express itself. It's just going to hang out. But if we have two recessive alleles, they both work together, and that's what we see. So we have some examples. Dark hair is a dominant allele over blonde or red hair, which would be recessive. So if you got one allele from mom that said dark hair and one allele from dad that said blonde hair, you would have dark hair because it is the dominant version of that gene. It will win every single time. So let's look at a couple other examples. Curly hair is dominant over recessive, which is straight hair. So that curly hair gene always going to win. So if you got, again, one curly hair allele from mom and one straight hair allele from dad, then you're going to have curly hair because it is a dominant trait. Widow's peak. Widow's peak is how your hairline is shaped. So widow's peak is a dominant allele. So if you got a single copy of a widow's peak gene, then you would have that widow's peak. So you can see upper left corner, you can see that picture shows what a widow's peak looks like, where it's pointed inward at the top of your hairline. Versus the recessive allele is a straight hairline, where it goes straight across. So widow's peak, again, that little pointy guy up at the top of your hairline, is dominant over the straight hairline. Freckles are dominant to no freckles. So again, if you have freckles, you have at least one copy of that dominant allele. Cleft chin. When you have a cleft chin, like right hand side has a cleft chin is when you have that little dimple in the middle of your chin. That is actually dominant over recessive. So if you do not have a cleft chin, that means that you have two recessive copies of that allele. So that means that you, again, have two recessive copies, no cleft chin. Dimples. So dimples are actually dominant. So if you have dimples, that you have at least one or maybe two copies of that dominant allele. But if you don't have any dimples, that means that you have two recessive copies of that gene, no dimples. So detached earlobes and attached earlobes. So you can see that lower left-hand corner. You can see the picture of the two options. So the attached earlobes are going to be attached to the side of your chin completely from top to bottom, where detached earlobes have a little hook and the earlobe is actually not completely attached. So you can check that out. Check that out on you. Check that out on your mom, dad, siblings, whoever you got hanging around, and see who has what. Because just because something is dominant doesn't necessarily mean that it's very common. So we're going to be talking about that more in the future with a survey that you're actually going to do. So those are just a few examples that you can check out. Alleles. So when we're talking about alleles, we're going to have to write about them and specify who has what alleles and who has the dominant alleles and who has the recessive alleles. In order to do that, we have to have letters. So dominant alleles we're going to represent by capital letters. So makes sense. They're dominant, they're going to be capital. Recessive alleles are represented by lowercase versions of the same letter. So if you're talking about one gene, you're going to choose one letter, and the dominant form of that gene, that dominant allele, is going to be that capital letter. So, for example, if we're doing freckles, if we want to say letter T for freckles, capital T would represent having freckles, that dominant allele, and then the recessive allele would be a lowercase t, because that would be, again, no freckles, recessive allele, so we're going to use that lowercase letter to represent that.
Hey guys, I just wanted to do a quick aside and talk about which letters are good to use to represent alleles when we are writing about these genotypes. One set, very good to use, is the letter F. Capital F and lowercase f look totally different, which you're going to need when you're representing dominant versus recessive alleles. Not such a great choice would be the letter O. O, capital versus lowercase, the only difference is size. So that's going to be very hard to distinguish when we are writing these things out, and we're going to be working with these charts called Punnett squares. And when the only difference is size for a letter, capital versus lowercase, makes it very hard to distinguish. Another example of not a super great choice would be the letter U. Again, only difference is size, capital versus lowercase. So some good choices. Letter T. Letter F, as we just talked about. Letter B is definitely a good choice. R, E, Q, A, D, or H are just some really good choices. Some alternative, not so great choices include, like we said, the letter O, letter S even, C, X, P, W, Z. The only differences between those capitals and lowercases is size. Those are not going to be the best choices. So I didn't go through the whole alphabet, but those are just some options for going forward.
So let's talk about these terms, phenotype and genotype. Phenotype is the physical appearance or visible traits. So physical, phenotype, they sound similar, so get those in your head to kind of cement that. Phenotype, physical appearance, versus genotype is the genetic makeup or which alleles an organism has. So genotype, genetic makeup, so they kind of sound similar. So genotype, genetic makeup. Phenotype, physical appearance. So your physical appearance can sometimes give you hints about what your genotype might be. So these are going to be very, very helpful terms going forward to figure out by looking at an organism what their genes might be. So, some terms to describe genotype. So we have these terms, purebred or homozygous. These have two of the same alleles, two of the same versions of a gene for a trait. So if they have two dominant alleles, example, they are purebred or homozygous dominant. Or if they have two recessive alleles, they would be purebred or homozygous recessive. So we have our example there. Purebred tall plants will have two alleles for tall stems. Two capital T's is how we represent that. While purebred short plants will have two alleles for short stems. And remember, our recessive alleles we represent with our lowercase letters, so two little t's for that guy. We can also have two different alleles. Those are called hybrids or heterozygous organisms. So they have two different alleles. So they're going to have one capital letter and one lowercase letter because they're going to have a dominant and a recessive. So in our example here, hybrid plants for height will have two different alleles, one for tall and one for short. So we have big T, little t. But if we have even a single big T present, if we have a single dominant allele present, that is the one that we will see. So this plant's phenotype, its physical appearance, would be tall. So what will be the phenotype? Again, that physical appearance. So first trait we're going to look at, dimples. Two alleles, two different versions of the gene that are possible. Are dimples or no dimples? Our dominant allele, always represented by a capital letter, is dimples. So having dimples is dominant. Capital D to represent that. Recessive allele would be having no dimples. I do not have dimples. So lowercase d to represent that. So what would the phenotype of a person with the genotype big D, big D be? I want you to take a second, think about that. What would the correct answer for that be? We have two dominant copies. If you said that they would have dimples, you are absolutely correct. Now, let's look at another one. We have a phenotype, or what would the phenotype of a person with the genotype, this is a hybrid one, big D, little d. What would happen there? Again, take a second, think about that one. So, again, we have a single dominant allele present. If even one is present, that is the one that we will see. So we would absolutely see dimples on that individual again, simply because that one capital letter, that one dominant allele is present. It will win every single time, no matter what. All right, so let's look at another example. This trait is going to be the color of a flower. So we have two alleles, two different versions of the gene. We have white or pink. The dominant allele in this situation is pink. We're going to represent that with a capital F, just like all of our dominant alleles, capital. Recessive allele is going to be white color. We'll represent that with a lowercase f. So now, let's take a look. What would the phenotype of a flower with the genotype of capital F, lowercase fb. So think about that for a second. See what you would think. Now, if you're thinking, okay, I see one dominant version of that allele, and that's all it takes. Dominant wins every single time. You're absolutely right. If we have a single dominant allele, that's the one we're going to see. So the dominant allele, pink, that's the one that's going to win. The flower's going to be pink. Now, we have a different situation in this next one. What would the phenotype of a flower with a genotype of lowercase f, lowercase fb? I'll give you a second to think about that one. Now, no dominant alleles seen here. So our recessive alleles finally get a chance to shine. There's no dominant alleles to kind of squash them down, so we're finally going to see that white flower with a lowercase f and a lowercase f. So how do we determine what alleles an organism has and what alleles the offspring could have potentially? For humans, we can do genetic testing. We have lots of really cool technology. A lot of families do genetic testing before they have kids, just to be aware of what potential diseases could be involved, not just physical traits. But we can also use old-fashioned Punnett squares to calculate what the parent's offsprings might inherit. So these Punnett squares, these are charts that show all the possible ways the alleles could combine in a genetic cross. So again, all the possibilities. First step to use a Punnett square is to identify the alleles of the parents. Let's take a look at an example. So we can examine how flower color is passed down between generations. Pink flowers, we just saw, are dominant, and white flowers are recessive. So we're using that same example again. Pink flowers, that capital F, white flowers, that lowercase f. We're going to cross a purebred, using this vocab term, purebred. I also want you to think, what was that other vocab term that also meant purebred? Give you a second. You said homozygous, you got it. So homozygous or purebred pink flower. So that means that purebred means has two copies of the same allele, so the same version. So that means it has big F, big F. While a white flower has to be little f, little f. Remember, even if it had a single copy of that big F, it would be pink. So if it's a white flower, we know it has to be little f, little f. So what would this Punnett square look like? First thing to do to fill out on a Punnett square is label these boxes with the alleles of the parents. We put the alleles of the parents on the top and on the side. So let's do that first. Parent 1 can go across the top. Parent 2 can go along on this side. So parent 1, capital F. And remember, so what we're going to do for the parents. Each allele is going to get its own box. So you're going to separate those two alleles, one per box. And then on the side, same thing, one per box. We're not going to smush them together or put them side by side. That's a common mistake. So make sure that you're looking at this right now. Parents, you're going to separate them one allele per box on the top and on the side. So parent 1, big F, each gets their own box. Parent 2, little f, each gets their own box. Now, we're going to look at all the ways that these parents could cross their alleles in their offspring. So first we're going to cross first box. Big F and a little f could combine to make big F, little f in the offspring. So these inside boxes each represent a possible offspring, what the offspring could possibly inherit. So one possibility, they could get a big F from mom and a little f from dad, and that could be their outcome. Now, we're going to do the same thing for that second box. We're going to take top and side. So now in this situation, we got the same outcome. We got a big F and a little f, so that offspring has the same potential outcome. We're going to do that same thing across the bottom. So parent 1, parent 2, coming down. And then again, last box, as you can imagine, same situation. We're just pulling them across from each other. So in this situation, all of the offspring possibilities are all exactly the same because the parents are both homozygous. So there wasn't very much mix-up. There wasn't many different possibilities. They're all big F, little f. Now, I am going to take a pause here. If we see big F, little f, what color are all of these offspring going to be? I'll let you think about that for a minute. If you said, they have a copy of that dominant allele always wins, they're all pink, you are absolutely correct. So, you have now created all of the potential offspring's genotypes inside the squares, created by crossing those parents. So, parent 1, parent 2. So, again, each new set of alleles inside those boxes represent a potential offspring and what their alleles would be. As I just mentioned, when we're crossing two purebreds or two homozygous organisms, all the offsprings are going to have the exact same alleles. That is always true. If you want, you can test it out. Make parent 2 two big Fs. Make parent 1 two little f's. You'll have the exact same outcome. One thing to note about Punnett squares, you'll notice that the big F always went first. Big F, if there is a big dominant allele, as you can imagine, they're always going to go first. No matter what, no matter which way we're crossing, who's what, when, where, why, the big F, the capital dominant allele, when we're writing out a genotype, will always go first. As you can see in each of those 4 boxes, that a capital F always went first. That will always be the case. Capital always goes first. So, I have a little reminder over there. Big F is pink, little f is white. So, what would a purebred pink flowers alleles be? I'll let you take a second. I want you to try and answer that yourself. Purebred, so think about what that means. Purebred means they have 2 of the same alleles. So, if they are purebred, they have 2 of the same alleles, and they're pink. What is their outcome? What are their alleles? What is their genotype? If you said big F, you're absolutely correct. Again, purebred means they have 2 of the same. Pink, we know that's represented by the capital F, so we have 2 of the same. Pink, boom, 2 big Fs. Now, what would a hybrid pink flowers allele be? So let's take a look. So hybrid. Hybrid means that they have 1 of each. They have mixed alleles. They have different alleles. So if we know that they have different alleles, they have mixed alleles, and they're pink, what is their genotype going to be? I'll give you a second to think about that one. So, if you said that we have big F and little f, you are absolutely correct. So hybrid, again, means that you have 1 of each. You have a mix. You have a combination of both of the alleles, both of those different types of the gene, or different versions of the gene, excuse me. So, big F, little f, hybrid. And again, because we have that 1 dominant allele, it wins every single time, so it has to be pink. Now, this question doesn't give you purebred, hybrid, heterozygous, homozygous. All it tells you is, what would a white flowers alleles be? Is that enough information? It absolutely is. I want you to think about it. If it's white, what does its genotype have to be? So again, think about that. If it's white, and that is the recessive allele, what does the genotype have to be? So, if you said little f, you're absolutely correct. Because remember, if you had a single, even 1 big F, 1 capital letter, that would make the flower pink. So it has to be 2 little f's. Now, this one's tricky. What would a hybrid white flowers alleles be? I want you to think about it, give it a try. Now, if you're breaking this down and you're going hybrid, that means it's a mix. It's two different ones. So if it has a big F and a little f, it has to be pink. This isn't possible. Then boom, you caught me. That cannot happen. It cannot exist. A hybrid white flower could not exist. The white color is recessive. Both alleles would have to be lowercase f in order for the white color to show. So it cannot be hybrid. It cannot be mixed. It cannot have two different alleles. Trick question. So that is the end of the show. Again, take a breath. If you have any questions, please let me know. Please go back, read the book if that's going to be a help for you. Definitely let me know if you have any questions. Feel free to email me. Feel free to post on that discussion board. Let me know in any way, shape, or form. It might be helpful to go back and watch this again. Please make sure that you are filling in your notes as well. I highly recommend printing them out, putting them in your science folder or your science binder, just like always, because there will be assessments on this. So please, please, please let me know if you have any confusion. We will be practicing with this a lot. We'll be practicing with these terms and how to make a Punnett square and how to go from offspring to parents in a Punnett square. So we'll be practicing with these a lot in the next several classes. But again, please let me know if you have any questions. Thanks.